In this session, I want to discuss about the DNA replication. So we all know the DNA as a carrier of genetic information from one generation to the another generation. So here as a carrier of genetic information, the DNA should have, or the genetic information material should have the following two functions. One is called heterocatalytic function and the second one is called autocatalytic function. So here in the heterocatalytic function, if the DNA directs the synthesis of chemical molecules other than itself, it means the synthesis of RNA and the synthesis of proteins, such type of function is called heterocatalytic function. The second one, the autocatalytic function. The autocatalytic function, it means if the DNA directs the synthesis of DNA itself. So that is called, such type of function is called it as autocatalytic function. So here in the DNA replication, it directs the synthesis of the DNA strand or the template strand, it directs the synthesis of DNA data. So here while coming to the definition of DNA replication, it can be defined as a genome duplication. It is nothing but the whatever the amount of DNA that is present within that particular cell, it will get doubled. So here the doubling process that can be only possible by the process of replication. So the main purpose of replication is for the generation of new cells. It should be the genome should be doubled. So here that process can be achieved by this mechanism that is called DNA replication mechanism. So here we all know the cell cycle or the different stages of cell cycle. The actual replication which takes place in the S phase. So we all know about the different phases that is called G1 phase, S phase, G2 phase and the M phase. So here in the S phase, the actual DNA replication which takes place within the S phase. So here, according to the Watson and Crick proposed double helical structure model, DNA structure model, so there are three possible ways of replications. They are called conservative replication, and the second one is called semi-conservative, and the third one is called dispersive. The first one that is called the conservative model. So the conservative model is nothing but after completion of replication, the data strands will be formed. So here, if the how they pair with one another basing on that particular um, observation, we can call it as these are the different models. Whereas in the case of conservative, the middle one, after completion of one round of replication, you can find the two parental strands will pair with one another, and the two newly synthesized strand they can be paired as a another duplex. So such type of mechanism is called as conservative. So whereas in the case of semi-conservative model, so if you observe the first one that is called after completion of replication, the one parental strand will pair with the newly synthesized one. So such type of mechanism is called as a semi-conservative. And the third one that is called the dispersive. Whereas in the case of dispersive mechanism, or the replication mechanism, the segments of newly synthesized and the parental strands will pair alternatively, so we can call it as a zigzag manner. Such type of model is called it as a dispersive model. So here most of the organisms they follow the semi-conservative replication. This mechanism was explained by the two scientists called Michelson and Stahl. So these are the two scientists. They have used the two principles for further to explain the semi-conservative model. So this is called the experiment. So here in their experiment they have used radiography, autoradiography, and the next one is called the centrifugation technique. So here for this they have used mass isotopes. So here in the mass isotopes they have used nitrogen. So normally in general the DNA they contain nitrogen in N14 form. So here the N14 is converted into the N15 by the supplementation of ammonium chloride or the heavy ammonium chloride, that is called N15 ammonium chloride, and they have incorporated into the media which is used as an ingredient. So by this, so the E. coli cells they have grown in the particular media for the several generations. Automatically the total N14 contained media that can be converted into N15. So here the accuracy of the isopicric density gradient centrifugation, that is, although the remaining substances they are the similar or the same, the difference in between the DNA, that is called N14 and the N15, they can separate it as a two different bands. So this is the N15 ammonium chloride and the N14. So here N15 is the slightly higher denser than the N14. That's why they form the lower band, and N15 will be the upper band, and N14 will be the upper band. So here after that, the whatever the cells they have cultivated within the N14 N15 containing media, they have transferred to the normal media. The E. coli cells they have transferred to the normal media. So after single generation of time, they have isolated the DNA from that particular E. coli cell and they have made centrifugation. So here, for example, if the process of replication which takes place as a conservative model, you can observe two bands. One is at the N14 region and another one is the N15 region. The one is N14 and the another one is the N15. So here replication which takes place as a semi-conservative model, a new band will be formed at the intermediate location. So here, so these are the three possible ways. So here after transferring of N14 culture cells into the normal media, after that they have isolated DNA and they are centrifuged. So here what they have observed is the middle one. They have not, after one generation of time, after isolation of that particular DNA, it forms an intermediate one. They could not find the band formation either at the N14 region and at the N15. So this is only possible because of the semi-conservative model of replication. So here this is the experimental evidence for the semi-conservative model of replication. So basing on this experiment, most of the organisms, so they have concluded that most of the organisms, the process of replication which takes place as generally they follow the process of semi-conservative model of replication. So thank you.